aguardar que dê início à nossa sessão. Alex, a hora que você quiser. Ok.
Can we start? Can we go? Yes.
Yes.
All right.
We start, Rodrigo. Thank you.
Okay, thank you, Luciana. So good afternoon. I'll be the mediator of the Cancer Biology Roundtable, which is a evolving topic, and there is still too much to learn about this disease. I'd like to start this talk with a quote from Peyton Rose, who is a Nobel Prize winner of 1966. He said that tumors destroyed man in a unique and appalling way, as flesh of its own flesh, which has somehow been rendered proliferative, rampant, predatory, and ungovernable. They're the most concrete and formidable of human maladies. Yet, despite more than 70 years of experimental study, they remain the least understood. He started his lecture of the Nobel Prize with these words, and I would like to add that more than 50 years after his speech, we are still learning about cancer biology. And unfortunately, we still have a long way to go. And we are here to discuss more about this topic, and I'm going to invite Professor Roger Chammas and Dr. Jõao Alonso to discuss it. First of all, we're inviting Professor Roger Chammas. He obtained his medical degree and his PhD at the University of São Paulo. He was a visiting researcher at Friedrich Miescher Institute in Switzerland, at Harvard School in the USA, at Federal University of São Paulo in Brazil, and at Moffitt Cancer Research Center in USA. He's a professor of University of São Paulo Medical School since 2000 and full professor since 2009. He's also a professor at the chemistry department of the North Carolina University since 2016. And he's the head of the Laboratory of the Experimental Cancer Research and coordinator of the Center for Translational Research in Oncology at the ICESP. He's vice president of the Deliberative Council from the Hospital das Clínicas, vice director of the University of São Paulo Medicine School, and member of the Director Council of ICESP. He has more than 200 publications, member of the editorial board of journals like Frontiers in Oncology, Cancer Biomarkers, and researcher CNPq-1A. His laboratory has been researching molecular biomarkers, extracellular vesicles, and focusing on strategies that enhance the effectiveness of affordable treatments, especially important in low-income countries. So, Professor Chammas, it's my pleasure to invite you to this roundtable.
Thank you, Rodrigo. Thank you so much for the kind invitation, and congratulations for the initiative of discussing pressing issues that we have in different areas of the biological sciences. I'm pretty sure this will be a very, very successful meeting, and hopefully to be the start of many interesting collaborations that we may have with you at Federal University. I'll share my slides. I think it was very fortunate that Rodrigo started with the quote from Peyton House. So it's true, we've been learning a lot from cancer biology in the last 50 years or so, but I want to point to that we need not only to understand more on about cancer biology, but we also need to understand what are the consequences of the therapy that we are afflicting to patients. And my presentation today will be an aspect of, indeed, this was a very surprising phenomenon we stumbled upon some years ago that is associated with the tissue response to therapy and how this tissue response can lead to failure in the treatment options we use. The idea here is, if you allow me, is to compare the treatment we do, for example, chemotherapy or radiotherapy, as the ego of Prometheus. As you may remember, Zeus tied Prometheus in a cliff, and every day an eagle would bite the of Prometheus, and during the night the liver would regrow. So this would happen for this time. I think that somehow the oncologists or the radiation therapists could be compared as the ego of Prometheus. And during treatment, they are able to
Recording in progress.
decrease the volume of the tumor burden, the volume of tumors, but during the interim intervals, what would happen is that tumors will regrow and causing what we call tumor cell repopulation of a disease that evolves. So the disease is not the same. So here we have a very, very serious problem to manage in the treatment of patients. Patient. So many years ago, we observed. Can you? I cannot go forward with the slides. Can you help me, please?
Dr. Chammas, I think that you have the control under your screen, maybe.
Okay, thank you. Sorry for that. So many years ago, we started doing experiments with engraftment of tumors. And one thing that we noticed as we started to observe what would be the very acute effects of tumor engrafting is that when we inject cells, either intramuscularly or in the subcutaneous tissue, one thing that happens 24, 48, 72 hours after the engraftment is that a large amount of tumor cells that we reject in mice, they would die. And the process of cell death here was associated to apoptosis. This is a tunnel labeling of cells that were injected in intramuscularly or subcutaneous of tissue of mice. So in the very first 72 hours after engraftment, what we see is a massive cell death. And cells which die here are not only the tumor cells which were injected, but eventually neutrophils that are recruited to the site of injection, they would also die. Based on this approach, we started to think, what would happen if we mix populations of cells that will be committed to apoptosis or committed to necrosis with viable cells, and what would happen if they would create a permissive microenvironment to tumor growth. And what we observed was that when we add necrotic cells to non-tumor or to tumor cells, we didn't have we had an attenuation of the growth of tumor cells. But if we added apoptotic cells, cells that are committed with apoptosis through ionizing radiation, we would promote tumor growth. So the lesson that we had from this initial experiments is that dying cells, when present in the same microenvironment of a very low dose of tumor cells, they can create conditions that support tumor growth. We started asking very fundamental questions. What are the materials that are made from this science? And we came across with a very interesting notion that if we take microvesicles, vesicles that are released by tumor cells in the process of cell death, these microvesicles alone, they favor metastasis, and we can block this experimental metastasis if we opsonize these microvesicles with an Exit5, which is a protein that binds to phosphatidylserine on the surface of the vesicles, impeding somehow the fate of these microvesicles. These microvesicles, they come in different flavors. These microvesicles, they carry different molecules. For example, they carry gangliosides. Gangliosides are glycosphingolipids that are decorating the outer leaflet of the plasma membrane. And extracellular vesicles having different contents of gangliosides. Specifically, these gangliosides are melanoma tumor markers, so they are enriched in melanoma cells. These gangliosides, when imparted in normal cells, they start in normal melanocytes, they increase the capacity of the cells, they increase the velocity of the cells in terms of cell migration. So they impart information to melanocytes that are somehow committed with a malignant phenotype associated with migration. Not only that, extracellular vesicles coming from cells that are dying through, for example, treatment with temozolomide, which is a chemotherapeutic agent that can be that has been used to treat melanoma patients before the emergence of drug therapies or immunotherapy. Temozolomide and cisplatin, two chemotherapeutic agents that have been used, they liberate their cellular vesicles from tumor cells. These extracellular vesicles can be quantified using different methods. Here we are using nanotrackings and nanotracking system to count these vesicles. These vesicles, they can incorporate into melanoma cells, modifying the behavior of these melanoma cells, but above all that, what they do is that the extracellular vesicles from treated cells, they interfere with they accelerate tumor growth, while extracellular vesicles from control cells do not do that. So they carry some information that change microenvironmental, the tumor microenvironment, favoring growth. And part of this information is associated with the induction of a macrophage imprinting towards what we call the protumoral phenotype. You are familiar with the notion of M2-like macrophages or tumor-associated macrophages that can improve the tumor growth. Extracellular vesicles from temozolomide-treated melanoma tumors shift the they have, as I mentioned to you, the phospholipids, and we are very interested in the metabolism of these phospholipids. For example, phosphatidylcholine is a very common phospholipid present in the membrane. Phosphatidylcholine is substrate of phospholipase A2, and which gives arachidonic acid and lysophosphatidylcholine. Arachidonic acid is further metabolized into different prostaglandins, and these prostaglandins can act as signals for a number of processes such as cell survival, proliferation, migration, invasion, antigenesis, and inflammation. The same way that another molecule that is derived from phosphatidylcholine, lysophosphorylate, which is called PAF, the platelet activating factor, which is produced by the acetylation of lysophosphorylcholine, which is released from phosphatidylcholine metabolism. PAF itself has the very same activities of this prostaglandins. So phospholipids can give different lipid mediators that will enhance tumor growth. We have a very interesting pharmacological strategy here to block PAF signaling, which is using PAF receptor antagonists such as WAB or CB series of molecules. It's important for the next experiments that I'll show you. The PAF interacts with a PAF receptor. It's as of today, there's only one member of this family of molecules that can be recognized with different reagents. PAF receptor is not ubiquitously expressed, but you see in tumors when we have hypoxic areas, which are these areas here, so I showed in red. We have induction of PAF receptor synthesis within hypoxic areas, not in all tumors, but in very selected areas where you have hypoxia. So PAF and PAF receptor activation seems to be associated with this low levels of oxygen, for example. We started to see if we could add some beneficial effect on adding PAF receptor inhibitors with the treatment of, for example, cisplatin, and I'll show you other strategies as well. So here's a melanoma growing in a nude mouse. As we treat the cells with cisplatin, we control 50% of the tumor growth. If we add WAB 2086, the PAF receptor antagonist alone, we have already this 50% effect as well. But if we combine both cisplatin and the PAF receptor antagonist, what we see is that we can control very efficiently tumor growth. We can control so efficiently that some tumors do regress, and we have up to 50% of complete tumor regression if we add both cisplatin and the antagonist of PAF receptor. So there is something that says to us that the PAF receptor signaling is important in maintaining a proliferating or the survival of the cells which proliferate even after treatment with cisplatin. When we published this set of results, another colleague from the University of North Carolina, Jõao, showed in a very elegant paper published in Nature Medicine that upon radiation or chemotherapy, they have shown really on radiation in this paper. They haven't done much work in chemotherapy. What they do is to induce apoptosis. Apoptosis would be associated with caspase 3 and 7 activation. Caspase activation would help, or would activate phospholipase A2. Phospholipase A2 would remodel phosphatidylcholine on the surface of the cells, releasing PGE2, prostaglandin E2, and extracellular prostaglandin E2 would be responsible for the phenomenon of tumor repopulation, which is the growth of the residual tumor cells that would then lead to tumor recurrence. We analyzed this phenomenon in melanoma, in different tumors, including melanoma. Here are the epithelium as a carcinoma. We observed that upon irradiation, there is an increase in BAF receptor accumulation or expression. Proteins accumulate here, and the message, the gene is transcribed, so there is increased expression of BAF receptor. And if we block BAF receptor signaling upon irradiation, we increase the effectiveness of the irradiation, of ionizing irradiation. So there is an effect of irradiation, as you see here in the white bars, but if we add the blocker of BAF receptor signaling, we increase this. So here we are clearly showing that there is an additional effect to radiation if we can actually block the receptor signaling. And then we repeated that very same experiment of mixing cells and looking at the favor and if we would favor tumor growth. And what we observe is that if we add irradiated tumor cells to luciferase expressing tumor cells, what we would see is that we would observe increase in IO8. There's a microenvironmental activation here. The cells would express more IO8. It's pro-angiogenic. And what we observe in the red lines is that the tumors which received the irradiated cells grew faster than the cells which did not receive the irradiated cells. And we could block this increased growth using antagonists of the BAF receptor signaling. And so we could control tumor volume using this strategy, decreasing the activation of BAF receptor signaling. So at that time, what we came with was this notion. We have production of BAF-like molecules that activate BAF receptor upon chemotherapy or radiotherapy. This would enlist signals that would favor cell survival and tumor repopulation. And at the same time, we have the notion that this BAF-like molecules could be acting on BAF receptor present in macrophages present in the microenvironment, and this will shift the balance, the programming of this macrophages towards an F2 phenotype. So this is the hypothesis we had when we decided to try blocking not only the signaling with a pharmacological inhibitor, but also the expression of these enzymes here, the lysophosphoryl acetyltransferases, that are responsible for the production of BAF upon different stimuli. The strategy we taken needed first that we need first to know which lysophosphoryl acetyltransferase is and how they are distributed in different tumors. So there are 4 genes. This is a 4-gene family of enzymes, lysophosphoryl 1, 2, 3, and 4. These 4 enzymes, I'm sorry, I messed up here. I should have another slide. This 4 enzymes, these 4 isoforms, they are differently expressed in melanomas, in different tumors. Most of the lysophosphoryl 1 is critically tumor associated, the others tend to be less associated with the tumors. So we needed to devise a strategy that would allow us to block the expression of the 4 lysophosphorylates, and hopefully would be doing this in a simultaneous fashion, because we really don't know how the expression of this different genes happen at the same time in cells. And to do that, we use an approach using RNA nanostructures. These RNA nanostructures are called nanorings. We can assemble these nanorings and set them in a way that we would add siRNAs to the structure of the nanoring. So we could have, for example, in an hexagon structured RNA ring, we would have we could have 6 different genes that are targeted at the same time in a given cell. So we started a collaboration with Terry Wafani, University of North Carolina in Charlotte, in the Department of Chemistry, and to design these nanostructures and use them to control the expression of lysophosphoryl acetyltransferases in melanoma cells. In the first experiment, what we noticed is that if we combine, for example, we use 3 silencing RNAs in the structure 1, 2, 3, we would be efficient in decreasing the expression of LPCAT 1, 2, 3 in the cells, but there will be a rebound effect and you start accumulating LPCAT4. This is why we need to try to devise a strategy at the same time silence, so we would not have a compensation effect through the silencing of a couple of them. So we started doing this, and we could show that if we add the ring with only the ring without only the ring without the siRNAs would not be able to present efficiently the process of cell death due to radiation as compared to the control. So this process of cell death of the hypodiploid cells were associated with increased expression, activation of caspase. So it's a caspase 3 and 7 dependent phenomenon, and this led to a decrease in the chromogenicity of melanoma cells. So we are increasing the efficacy of irradiation through the use of blockers, specific blockers of lysophosphoryl acetyltransferases 1 to 4. This lead us to this figure here that is an abstract, and it's a way that we are understanding the process now. We have chemo radiotherapy acting on tumor masses. This will change the production of magnets, which now we can control using these RNA structures. And if we do that, we block the process of tumor repopulation and increase the effectiveness of treatments to radiation and eventually to chemotherapy. With that, I would like to show you Renata Saito. Renata Saito was, she's a researcher in our group, and she's responsible for most of the experiments done here with the help of Maria Cristina Rangel, Luciana Nogueira de Souza Andrade, and Silvina Bustos. I would like to stop here and invite you all to come visiting us at the Cancer Institute in São Paulo, where we have our lab. Thank you, Rodrigo.
Thank you very much, Professor Roger Chammas. It was a really nice lecture. It was just a piece of the amount of published papers that you have that I know. But thank you very much for illustrating it so clearly. Now, I'm going to invite Dr. Jõao Alonso. After Dr. Jõao Alonso, he has his medical degree at Jundiaí School of Medicine. He obtained his master's degree and his PhD at University of Campinas. And he's a full member of the Brazilian Society of Urology and attends and coordinates the urology service at Paulínia Public Hospital. And he has published in really high impact journals like Journal of Clinical Oncology, that has an impact factor of 44, it's really good. And he's currently investigating the immunotherapeutic agent called oncothorate. Okay, so thank you. And after this, we will be opening for questions. If you have any questions regarding his work or professor, you can watch it here and we'll bring it during the session after Dr. Jõao Alonso's speech. So please, Jõao, you there?
Good afternoon. My name is Jõao Alonso. I'm here to present you a new immunotherapy to bladder cancer called the oncothorate. I would like to thank INFESP and all the organizers of this event. Congratulations, Professor Roger Chammas, for your brilliant presentation. It is been an honor to be able to participate. I also thank NCAMP and my group, especially the nanotechnology and medicine, a fast and safety of oncothorate immunotherapy in Bacillus calmette bearing responsibly non-muscle invasive bladder cancer. Nanomedicine is the medical application of nanotechnology. It uses nano-sized tools for diagnosis, prevention, and disease treatment. The ultimate goal is to improve the quality of life. Today, I will present the use of nanotechnology for the treatment of bladder cancer. There are two types of immune system, innate and adaptive. These systems are expressed by various mediators and modulators that are activity in the tumor microenvironment and homeostasis between the metabolic and regulatory system. And the system has certain phenomenon and inflammation. The inflammation can promote immunogenesis or prevent it. These effects depend on intensity and nature of the inflammatory response. If chronic inflammation, we will have a proliferative and carcinogenic. And if acute inflammation, we will have an apoptotic pathway. The immune system recognizes and destroys tumor cells through activated T lymphocytes and and other mechanisms. Tumor cells express several antigens that are not expressed in normal tissues. Immune system and checkpoint. The checkpoint inhibitors unmask a tumor cell. Since 2011 until today, the interest and the use of treatments has only grown. Two researchers won the Nobel Prize in 2018, an American, James Ellison, and Japanese, Tatsuko Honge. The first are studies of CTLA4 molecules, and the second for the development of anti-PD-1 components. The PD-1 pathway can be explored to evade the immune response. Tumor cells can reduce the lymphocyte activity through the PD-1 immune checkpoint pathway, expressing PD-1 and TL2 ligands. In a review of 25,000 articles, the most important topic in innate immunity research is dendritic cells, probably due to the ability of these cells to induce primary immune responses and connect the innate and adaptive immune systems. In this sense, activation of the toll-like receptor pathway is identified as a potential strategy to stimulate anti-tumor immune response. TLR2 and TLR4 receptors are best studied due to the wide variety of cells that expresses them. Cells have transmembrane receptors and recognize bacterial patterns and trigger an inflammatory response. Immunological sensors are distributed in the cells of the immune system. But what the nanotechnology, immunotherapy, BCG, and bladder cancer have to do oncotherapy? Oncotherapy and BCG are immunotherapies that treat blood cancer. Oncotherapy is a immunotherapy. Let's understand how they act. Bladder cancer has a higher incidence in men, almost 3 cases to 1. Second most common urinary tract neoplasm, 9th most incident type in the world. And by the National Cancer Institute in Brazil, in 2020, we had 10,600 new cases, claiming the incidence of 3 men to 1. According to the American Cancer Society, there were 81,400 new cases and also with high mortality. Smoking is the most important cause of bladder cancer, 70%. It was common in late 55 and higher incidence in men and white race. The trajectory of the patient with bladder cancer is very long and hard. From the appearance of blood in the urine, persistence, uncertainty in diagnosed, numerous tests until diagnosis, followed by extensive and difficult treatment. And after a phase of induction and maintenance of BCG 1, 2, 3 cycles, control tests, tumor recurrence, until culminating in conservative treatment or cystectomy. All of this generates a lot of costs and emotional suffering. For the diagnostic of bladder tumor, the main symptom is hematuria. An ultrasound and cystoscopy should be done, as we can see. Urothelial carcinoma is the most frequent histological type. Here, the ultrasound is usually the first imaging exam requested, as we can see here. Then on slide A and B, transurethral resection of the bladder as a form of biopsy and treatment. 17 to 75% are non-muscle invasible. Histological staging depends on the depth of tumor invasion in the bladder wall. And BCG. In 1902, we have the first strain of Mycobacterium bovis. In 1906, Albert Calmet and Camille Guérin observed the immunogenicity during its cultivation. In 1924, Pasteur began a massive production of the oral BCG vaccine. But it was in 1976 that Dr. Álvaro Morales and colleagues conducted the first clinical trial of intravesical BCG, observing a reduction in recurrence rates of superficial bladder cancer in 9 patients. And since 1973, the UFPAIVA in Brazil, Rio de Janeiro, BCG has been producing. OncoBCG is the gold standard immunotherapeutic treatment for non-muscle invasive bladder cancer. However, the treatment is empirical and limited by failure. Two-thirds of our patients suffer from adverse effects and treatment intolerance. Only 16 finish treatment within 3 years. It promotes response in around 70% of cases, but recurrency in 40 to 70% in 2 years. A big problem in the world today because significant unmet medical need in non-muscle invasive bladder cancer. 430,000 new cases each year globally. The scarcity of BCG complicates the treatment of patients. And after BCG, the patient's only option is undergo radical cystectomy or face cancer progression. The radical cystectomy remains recommended as a treatment option after BCG failure. However, 60 to 70 lifetime risk associated with non-muscle invasive bladder cancer cystectomy. Significant rates of morbidity, 30 to 60% in 90 days, and mortality, 2 to 9% in 6 months. Complication rate of 64% in 90 days, and 35% of patients require emergency room visits, and 26% require readmission. In Brazil and abroad, BCG is always in shortage. This compromises the treatment of patients with bladder cancer, mainly because it is an immunotherapy that can't be interrupted. What do after BCG failure? Radical cystectomy or bladder preservation therapy? We need to consider quality of life, surgical morbidity or mortality, recurrence risk, and patient decision. Now, I'd like to introduce you a new immunotherapy, oncotherapy, genuinely Brazilian, developed at the Unicamp in phase 1-2 of clinical research. I've been treating patients with bladder cancer. Oncotherapy is the fusion of an inorganic complex with a protein, forming the oncotherapy compound, with at least one of its sites in nanoparticle. The oncotherapy is patented in the United States, Europe, and Brazil. Thinking about immunotherapy, we performed a human clinical trial denominated Effects of Oncotherapy Intravesical Immunotherapy in the treatment of patients with relapsed non-muscle invasive bladder cancer, non-responsive to Bacillus calmette-guerin BCG therapy. This study was approved by Unicamp's Ethics Committee, registered by the Brazilian Registry of Clinical Trials, and supported by government assistance from FAPESP. Patients included weekly intravesical stimulation and intramuscular injections, initially for 6 weeks and then followed by an application every 2 weeks for 3 months and once a month application until the end of treatment, 24 months. Follow-up was performed with ultrasound and cystoscopy with systematic mapping of bladder biopsies. The primary outcomes were pathological complete response and recurrence-free survival, and the secondary outcomes were time to disease recurrence and safe response. This is the design of the clinical trial with 36 patients. All the patients were initially submitted to transurethral bladder resection performed by me at the public hospital of Paulínia, São Paulo, or at the patient's home service. Here the inclusion criteria and exclusion criteria. As for the demographic and general characteristics of patients, we have age median 65, 65.5% chronic smoking, 86.2% at least 2 previous BCG cycles. And what about the BCG failure? 65.2% were refractory and 37.9% relapsing. And 7 patients had used intravesical chemotherapy. Before oncotherapy, patients had an average of 2.2 recurrency in 24 months. And with oncotherapy, this average reduced to 0.2. Comparing patients treated with BCG and then with oncotherapy, we can see reduction from 29 to 3 patients with relapsed grade tumors in 24 months, reduction from 26 to 1 patients with tumors larger than 3 centimeters, complete response of 23 patients out of 29, and median recurrence-free survival of 22.2 months and at 24 months of follow-up. And here the complete pathological response was 100% up to 9 months of treatment, and 89.6% at 12 months, and 79.3% at 24 months. By the Kaplan-Meier curve, the median recurrence-free survival was 22.2 months. Mean time for disease recurrency, 20.6 months, and the average duration of response, 15.3 months. And about adverse reactions, skin rash is more incident, and just 13.8% grade 3 to 4. And no patients needs hospitalization to correct adverse reaction. Here are just a few examples of ultrasound exams and the cystoscopies and histological slides before and after oncotherapy. post-treatment evolution of carcinoma in situ and high-grade tumors, processing to chronic erosive cystitis with granulation and follicular cystitis. We note here the presence of a lymphoid follicle presenting more than 50% of the patients treated. What would be the role of this lymphoid follicle or the relationship with the therapeutic response? It is evident in these follicles a greater expression of interferon inducers when compared to normal tissue. Then a connection of the innate and adaptive immune systems through dendritic cells. We can see here the increased immunoreactivity of the markers before and after oncotherapy, such as TLR4, enos, and interferon gamma. And now, the mechanism of oncotherapy. We have two forms. The canonical form through TLR2 and non-canonical form through TL4. Oncotherapy produces a distinctive activation of the innate immune system mediated by TLR2 and mainly TLR4, resulting in increased signaling pathway for interferon, with interferon induction supported by TBK1, with activation of the STING complex, TROG phosphorylation and translocation to the nucleus. In the effective, cytotoxic effective pathway, there is recruitment of M1 macrophage or polarization of M2 to M1. M1 produces cytotoxic enos and activities and key directly in the site, producing cytotoxic perforins, or indirectly, hematologic, stimulating dendritic and NK cells, producing an activation of cytotoxic T lymphocytes, CD8, and tumor cell DIF. In addition, tumor cells are inhibited by interferon gamma, which binds to its receptors present in M2 macrophage and Treg lymphocyte, responsible for proliferation, chemoresistance, and metastasis. Interferon gamma receptor inhibits RANK and RANK L, which is the nuclear factor interleukin KB receptor and the proliferative pathway and metastasis. Here, an example of anatomopathologic result, a recent exam of a patient who had relapsed after BCG and radiotherapy using only oncotherapy for 2 years. As a result, chronic cystitis and the trusor muscle with fibrosis. Is onco-BCG still the best option for treating non-muscle invasive bladder cancer? Dr. Álvaro Morales, inventor of onco-BCG, recently published Bacillus calmette-guerin intravesical has been the gold standard immunotherapy in the treatment of high-risk non-muscle invasive bladder cancer for over 4 years. But he admits that inability of BCG to activate the STING pathway and limits the immune activity and anti-tumor response of BCG. Some considerations and comparisons of BCG and therapy. BCG, being biological immunotherapy, is only tolerated by bladder instillation, and oncotherapy can be used in the bladder instillation and intramuscular instillation, intramuscular injection. Sorry. As for the characterization of the onco-BCG is composed of live attenuated strain of Mycobacterium bovis, and oncotherapy a synthetic nanocompound and less adverse reactions. Activation of the immune system by onco-BCG is by TLR2. It only stimulates innate immunity, fails in 40 to 70% in 2 years, and oncotherapy acts on TLR2 and mainly TLR4 pathways, and induces interferon gamma, inhibiting RANK and RANK-L, and fails 20.7% in 2 years. Only 16% of patients complete 3 years of treatment with BCG, and patients completed 2 years of treatment with oncotherapy and without loss to follow-up. BCG is the most effective form of intravesical therapy. However, there is a production failure and compromise the patient's treatment. Then oncotherapy can be effective in BCG failure. In conclusion, oncotherapy appears to be a safe and effective treatment option for patients with high non-muscle invasive bladder cancer. It might provide benefits in preventing tumor recurrence and sparing cystectomy. On here, Dr. Wagner and Dr. Nelson Duran, the great inventors of oncotherapy, and our team. I'm so grateful and blessed to be part of this team. I also thank the patients for trusting our work, and special thanks for my wife, who is not in this picture, but he helped me in preparing this presentation. Thank you.
Thank you very much, Dr. Jõao Alonso, for such a nice presentation, and congratulate to you and Professor Wagner Pavaro for developing such a nice treatment option, because we have known considering bladder cancer, and it's nice to have an option, therapeutic option, and national therapeutic option. So we are opening for questions now. I think the first question was from who here? Caroline Ferreira. She asked, yeah, it can go to either one of you. Why is the incidence of cancer in men higher than in women? Okay, just Jõao or maybe Roger, both can answer it.
Professor Roger.
So I'm not sure if the incidence is that different. Do you have these numbers, Catarina, right? Or what is
Carolina.
Carolina? But honestly, there is no. Well, we can imagine different. Okay, the question is for Alonso. Probably she's talking about bladder cancer.
Jõao, how is the statistics for bladder cancer? Is there a higher incidence in men?
Sorry, Rodrigo, I don't understand.
For bladder cancer, the incidence is higher in men?
Yes, is more incidence 3 to 1, men to woman. The smoking is the main factor, 17%, and the incidence is higher in men than women.
Thank you very much, Dr. Jõao. Okay, the next question was from Ileana Sanchez Rubio. She asked to Dr. Alonso, why did your administration of oncotherapy intramuscular, while BCG is just intravesical?
Rodrigo, posso?
Yes, okay, you can speak.
Posso falar em português? Você pode repetir a pergunta, por gentileza?
Por que a administração da oncoterapia também é intramuscular? Vocês fizeram intramuscular e não só intravesical.
Isso. O fato de ser um composto inorgânico associado a uma proteína, uma nanopartícula, e não se trata de um imunoterápico biológico como a onco-BCG, que traz bastante reação e reações adversas e até pode provocar doenças como doença pulmonar por tuberculose ou em outras partes, como eu já peguei em epidídimo. Então, essa imunoterapia, por ser sintética ou semissintética, ela pode ser aplicada por via intramuscular sem trazer reações adversas, por não se tratar de um tratamento biológico, principalmente.
Tá legal. Thank you, Dr. Alonso, for your answer. There are several messages here from Luciana Caperuto, Maria do Céu, Marcelo Moura. They are all congratulating both of you for your nice presentations. Patricia Sartorelli also congratulated both. She said, thank you, Dr. Chammas and Dr. Alonso, for excellent talk. My question goes to Professor Roger. Some possible drugs have, instead of a cytotoxic effect, an immunomodulatory effect, triggering events such as some beta, with the production of NO or IL-6 such as some beta-glucans. In this case, is this related to the surface of the macrophage or more to the characteristic of the substance?
Well, thank you all for the feedback. Patricia, beta-glucans are recognized by a family of receptors that were alluded to in the talk of Alonso. Beta-glucans do interact with toll-like receptors, and this would activate cells which express toll-like receptors. Macrophages, cells from the innate immunity, are very rich in toll-like receptors, but not only these cells express toll-like receptors, other cells as well. Epithelial cancer cells also express beta-glucan receptors, such as toll-like receptors. So this will depend then into a feature of the molecule, the beta-glucan, and a receptor which is expressed on the cell surface of macrophages and other cells as tumor cells. And this activates, it causes a pro-inflammatory environment. Now, if you have death, cell death, within a pro-inflammatory environment, then you can elicit an immune response that will fight the tumor. And this is what Alonso showed very nicely and very elegantly with Oncotherapy. So this would be a possible mechanism, okay?
All right. Thank you very much, Dr. Roger. Maybe going along with this question, talking about immunomodulation, considering tumor heterogeneity, how would PAF-R inhibitors will be affected, considering there are this heterogeneity, and maybe you're combining it with radiotherapy? Would you like to combine with other therapies that would like induce immunogenic cell death, or I don't know, something that would have a broader effect? What do you think about?
Yeah, you see, there is very strong evidence now that radiotherapy would not work if you don't have production of interferon, type 1 interferon, which also, I mean, it brings us very closely to the theme that Alonso showed. So you need to have signaling through the interferon response. You need to have interferon response to have an effective radiotherapy treatment. So does that answer your question?
Yes. Okay. Thank you, Professor Roger. Another question from Carolina Ferreira, again for Dr. Alonso. Is the urinary microbiome associated with bladder cancer? O microbioma urinário está associado com câncer de bexiga?
Sim, está associado. Cistites crônicas, por exemplo, podem levar ao desenvolvimento de tumor de bexiga, né, por alteração, desarranjo da parede celular, degranulação de mastócitos e assim por diante, por cistites de repetição, cistite actínica, cistites intersticiais e agentes externos como uso de tintas, anilinas, radioterapia e vários outros fatores que podem mexer com o microambiente tumoral vesical e realmente favorecer o surgimento de um câncer vesical.
Mm-hmm. Considering microbiome, we are talking about it. Gut microbiome has an effect in immunotherapy, like checkpoint blockade, PD-L1. It affects how it works. Do you think that it has also an effect on Oncotherapy? O que eu estou querendo saber é, considerando o microbioma,
Sim.
isso também pode estar afetando a funcionalidade do Oncotherapy?
Posso responder? Pode. O Wagner vai responder.
Posso responder? Bom, esse é um assunto importante. Definir, né, caracterizar o microbioma da bexiga é um dos projetos nossos, um grupo que já trabalha com microbiota intestinal aqui na Unicamp, grupo que trabalha com diabetes, justamente para nós identificarmos se isso tem alguma influência ou não. Mas um dos componentes, né, o componente proteico do oncoterade, ele acaba tendo uma ação bactericida. Provavelmente a ação, né, no microbioma pode ser que ocorram algumas bactérias que estejam, né, em algum micro-organismo que esteja presente na bexiga, e esses fragmentos possam estimular localmente toll-like no urotelial. Então, nós estamos estudando um pouco da questão até de uso de pré ou probióticos na modulação de onco-BCG, da resposta de onco-BCG. Uma das hipóteses é que o microbioma pode ter uma interferência na resposta de onco-BCG. Ou seja, a resposta em manter uma resposta TH1, né, sustentada, pode ter influência do microbioma, interferir nisso. Então, algo que nós estamos tentando caracterizar primeiro o microbioma. Muitos desses pacientes, por exemplo, que têm câncer de bexiga, eles têm uma série de infecções urinárias de repetição, com E. coli uropatogênica, que está relacionada à progressão do câncer de bexiga. E muitas vezes esses pacientes com E. coli resistente têm dificuldade em responder ao BCG. Então, esse é um fator importante de nós estudarmos o microbioma e até mesmo interferir para que drogas ou terapias que vão modular o sistema imune possam ser mais efetivas. Então, essa é uma ideia que o grupo persegue. Não é tão simples assim desenvolver. O professor Roger Chammas conhece bem isso. O microbioma, ainda no câncer de bexiga, o intestino se avançou muito, né? A microbiota intestinal, mas na bexiga ainda nós estamos caracterizando, né? Em modelos de roedores, nós já fizemos isso, mas é um pouco complexo, porque é um microambiente totalmente diferente para a caracterização do roedor, né? Em lagomorfo, que são os coelhos, mas agora nós começamos a fazer isso em seres humanos, né? Principalmente esses pacientes que frequentam o laboratório do Dr. Jõao Alonso, para nós caracterizarmos um pouco isso, tanto na biópsia, tentar tecidual, quanto na urina. Nós temos um grupo aqui dentro do CNPEM, que também nos ajuda a fazer essa caracterização do microbioma.
Okay, thank you very much,
Obrigado.
Professor Wagner and Jõao Alonso. I have another question from Anderson Bueno. He said, amazing lecture, Professor Jõao Alonso and Roger Chammas. I have a question to Dr. Jõao. Does the BCG have any role on other types of tumor? I extend my question. Do oncoterad has any action on other kinds of tumors?
Outros tipos de tumor.
Pelo que eu entendi, se a gente pode usar o oncoterad para outros tipos de tumores também, é isso?
BCG ou oncoterad?
Bom, BCG ele é usado apenas em relação a tumor, apenas para tumor de bexiga, né, que pelo menos que eu tenha conhecimento. O oncoterad a gente tem feito uso para tumores sólidos não hematológicos em outros protocolos que já desenvolvemos. Inclusive, tem outros trabalhos em andamento, já autorizados em comitê de ética para tumor de cólon, mama, que a gente já está com vários pacientes em tratamento. E para tumores sólidos, outros, como esses que eu citei, a gente tem tido bom resultado também, assim como na bexiga. E a associação, talvez a pergunta da associação do Oncoterad com OncoBCG, nós não temos experiência. Quando começamos o tratamento, inclusive estava em falta OncoBCG, a gente até desenvolveu esse tratamento com um braço único, porque não tinha disponibilidade de BCG. Então, nós comparamos todos os pacientes que chegaram com tempo médio em torno de 10,8 meses de recidiva com BCG e pelo menos tendo tido 2 ciclos de tratamento. E com o Oncoterad, durante 2 anos, a gente percebeu que houve uma diferença muito grande em relação ao tempo de recidiva e ao tamanho do tumor e ao número de recidivas. Então, assim, nós não tivemos ainda, não podemos comparar ou usar junto com o BCG o Oncoterad.
Tá legal.
Existe, se você pode me deixar complementar, Rodrigo.
Desculpa, não, eu achei que você tinha terminado.
Nós temos tido, nós estamos usando em alguns pacientes um protocolo compassivo de pacientes compassivos, a associação do Oncoterad com alguns inibidores de checkpoint, e essa associação parece ser positiva.
All right. Thank you very much, Dr. Jõao. I have a question from Professor Ileana Sanchez Rubial. She's asking Dr. Chammas. Dr. Chammas, are there clinical studies using PAF-R inhibitor together with other chemotherapy, radiotherapy?
Yes. There's a molecule called rupatadine. Rupatadine has been used, well, it's not a clinical study, but it's a suggestion that may be one day a clinical study for ovarian cancer. We tried rupatadine in radiation therapy. We didn't have success in its use, though. But eventually in ovarian cancer, it may be a different ballgame. I'm not sure. So, but people are looking at different possibilities. The one problem is that these molecules aren't they are not clean PAF receptor inhibitors. They may have different functions other than inhibiting PAF receptor. If I may, Rodrigo, I would like to add something to the discussion of BCG and cancers. I think Anderson was. Many years ago there was there were studies addressing BCG in melanomas as immunoadjuvant. This really anticipated an area of immunotherapy for melanomas, which now is nonsense because we do have the immune checkpoint blockers, which are very specific. BCG is, as you all know, is the whole bacteria, and we use BCG for immunization against tuberculosis. So, as Alonso mentioned, there was a time that we have paucity, we have shortage of BCG for immunization of tuberculosis. So we need strategies such as Oncoterad that makes the solicitous replaces OncoBCG. So it may not be the case anymore to have a very complex mixture of bacteria to induce an immune response. If we can purify the immune inducer and use them in a very controlled manner, we are in pretty better shape than using OncoBCG. So hopefully, and as Alonso mentioned, only cavity tumors would benefit from BCG today, and only vesical tumors are. It's the only clinical indication, and we need to progress towards something that has a very well-defined composition. BCG is impossible to control, but molecules that are complex mixtures are semi-synthetic molecules as oncotherapy can have quality control. And if they do have the very same effect of inducing interferon response, this is what we need to go ahead as an immunoadjuvant, right? So I think indeed I would like to congratulate Alonso and all the group Fábio for the vision of developing such a smart strategy. It's very, very interesting to have this development here in Brazil.
Yeah, very nice.
Thank you, Dr.
We can establish now a link between you and Alonso and start using PAF inhibitors and Oncoterad together, maybe combining both strategies and see what happens.
Yeah, very good.
The commission has congratulated you both. She asked the question, but I think that it has been answered by Dr. Alonso. Diego Zandona is very satisfied to see Professor Wagner. He said that he worked with you anatomy in 2017. Okay, so just saying hello. Luciana Capiroto just said thank you very much for your researchers in this area, Dr. Chammas and Dr. Alonso. Especially when we think about the patients that will be beneficiaries from your work. Do we have any more questions now? Well, both presentations have been good, excellent. We still have some time to discuss other subjects. Professor Roger Chammas, just a question that doesn't have anything to do with the subject, but you have been in the board of Hospital das Clínicas and ICESP, and what's going on with clinical trials and everything else during COVID pandemics? I mean, what's going on there, and how is going scientific research, how it was affected, and how it's our future? What do you think will happen? I mean, just a broad question, but I think it's a relevant topic that we can ask.
So there was clearly a slowdown, not only in clinical trials, but unfortunately in patients refrained to come to the Cancer Institute because of the pandemics. We had a considerable delay in diagnosis. So we are facing today the challenge of having more and more advanced cases than we had 2 years ago because of the delay we saw. For the clinical trials themselves, whichever clinical trial was on, it kept on. We had, as a safety measure, we avoided to take samples from patients before remodeling all the area to process the samples in a safe way. So there was a slowdown. Now, my concern, I mean, the clinical trials will now ramp up again. I'm pretty sure that will happen. We learned a lot with telehealth and use of telemedicine. approaches, and we are now including these telehealth approaches. And I think they will ease, this will be the positive legacy of the pandemics. We will be better, in a better shape using telemedicine. And this will also help for contacting patients for research purposes as well. So this is the positive side of the pandemics. The negative side of the pandemics is that we are economically broken. And the federal government is broken and the state government is broken. It's not broken, but it's securing money. So we will face some difficult times, especially regarding the maintenance of equipment that are and so we probably need to talk more, and the scientific community should try to have to come to the agencies, and together with the agencies create a positive environment for research and stimulate share use of equipment and rationalize our research efforts, because there will be shortage of money. And better than we understand that we can collaborate, and I think we need to come with this conclusion together as a community, and avoid those initiatives that would put us competing against each other. Otherwise, we won't be effective in doing long-term research anymore. I'm really worried about our future as a community, especially based on the science of the federal government. So I think we need to come together and arrive to get a consensus on what to do, how to do, and present it to the executive, our representative, the executive representatives. This is my opinion. Thank you for letting for the opportunity to speak it out.
Thank you for talking about it. It's a very important topic, I think. How Dr. Alonso, how was the COVID-19 impacting your clinical trials, or how it was in the clinics? Alonso, how foi o impacto da Covid-19 na sua clínica e nos clinical trials?
Um impacto muito grande, né? Já que no início, principalmente em relação à vida privada, a gente consultório fechado, os hospitais, tanto o serviço privado quanto o serviço público, uma restrição de atendimento bem drástica, né? bem alta. E em relação ao estudo, ao trabalho, a gente conseguiu caminhar com o trabalho, manter o trabalho, restringindo a aglomeração, conseguindo convidar os pacientes para vir ao ambulatório em horários diferentes, ampliando o atendimento, ficando até mais tarde para atendimento, ou seja, um desdobramento de equipe, desdobramento pessoal grande, não só nós, como dos próprios pacientes. E, além de tudo isso, principalmente a gente que trabalha no SUS, lida com o SUS, a gente notou muito a deficiência que tivemos e temos, né, em termos de acolhimento de materiais como máscara, utensílios básicos, que muitas vezes o SUS sofreu demais em poder colocar isso à disposição. E eu acho que foi um grande aprendizado e percebemos, sem dúvida, que o país sofreu com isso pelo despreparo, talvez seja essa a palavra certa. E, como sempre, para quem faz ciência, para quem traz desenvolvimento de clinical trials, de trabalhos, acaba pagando dobrado, né? Porque a gente, os fomentos, as empresas governamentais que nos ajudam, que bancam os trabalhos, já é muito difícil você conseguir alguma coisa, e ainda mais quando tem uma pandemia, tudo piora. Então, foi uma grande experiência desagradável, mas um grande aprendizado. O Wagner quer fazer uma consideração?
Só uma consideração. Tem um fato que foi muito importante, pelo menos no estudo conduzido pelo nosso grupo, em especial o Dr. Alonso, muitos desses pacientes, nós fizemos no Brasil hoje uma condição médica não atendida para câncer de bexiga. O Roger falou muito bem com relação à produção de um PCG, não é fácil, a qualidade dos lotes de produção, a reprodutibilidade desses lotes de produção, o fornecimento ele é descontinuado, não só como na Europa, como nos Estados Unidos. Então, esse paciente que é diagnosticado, ele faz uso, muitas vezes, de quimioterapia, que não tem uma evidência de funcionar esse paciente muito bem, e muitas vezes o urologista opta apenas pela ressecção transuretral, né? E esses pacientes, por não ter, né, acesso a essa medicação, ou muitas vezes, né, pela pandemia, não conseguir ter um acolhimento rápido, ter um diagnóstico, paciente fria com hematúria, foi tratado, muitas vezes mal diagnosticado, mal conduzido. Quando esses pacientes chegavam para a triagem, para o estudo, eles já tinham uma doença mais avançada. Então, esse foi um ponto que dificultou bastante, porque eram pacientes que, no início, quando eles tinham uma ressecção transuretral prévia, quando chegavam para a triagem, o Dr. Jõao tinha que ressecar de novo esse paciente, e nessa nova ressecção, muitas vezes o tumor já era um tumor músculo invasivo. Então, e isso com uma questão de poucos meses, 3, 4 meses após a última RTU. Ou seja, uma doença que muitas vezes era uma doença de baixo grau, pelo fato de não ter tratado com o BCG, esse paciente, quando tinha o urologista, encaminhava esses pacientes, nos encaminhavam pacientes para incluir no protocolo, já eram pacientes de falha, mas quando eles vinham, né, com o encaminhamento do urologista, com a doença ainda dentro dos critérios, nós avaliávamos novamente esse paciente, e muitos desses pacientes não tinham mais critério para inclusão, porque a doença havia progredido. Então, isso foi um impacto muito grande. Então, assim, a pandemia para o câncer de bexiga foi terrível, porque muitos pacientes não tinham tratamento com o PCG, muitos pacientes não conseguiam chegar ao serviço, né, especializado, um ambulatório de oncologia, um ambulatório de urologia, para serem triados e diagnosticados corretamente. Então, nós tínhamos um atraso, né, na vinda desses pacientes e também a questão de não ter acesso à medicação. Enfim, esses pacientes, muitos tinham acesso, já tinham feito um PCG, mas o problema é que haviam falhado e é uma nova tentativa de se fazer, né, um PCG. Então, nós tínhamos um cenário difícil para câncer de bexiga aqui no Brasil, né? Ainda mais considerando todas considerando todos esses cenários que a pandemia nos mostrou. O câncer de bexiga, infelizmente, no Brasil, é uma doença subnotificada. Vocês podem comparar os dados, né, de estatísticos em relação à sociedade americana para o câncer, 80 mil novos casos por ano, e o Brasil 10 mil novos casos. Talvez não seja igual, né, ou próximo, mas que é muito agravou. Então, o apelo à ciência é justamente isso. Eu acho que no Brasil, acho que nós conseguimos construir a estrutura para os ensaios clínicos no Brasil, muito bem montado, para atender, muitas vezes, a demanda da indústria farmacêutica ou de protocolos que já vêm desenhados. E agora é o nosso papel, né, como cientistas e pesquisadores envolvidos em ensaios clínicos, de desenhar, né, propor novas soluções, propor novas estratégias terapêuticas, não só medicamentos, mas reposicionarmos medicamentos, desenvolvermos nossas estratégias e até mesmo desafiarmos essa própria estrutura, né, que foi montada por nós mesmos de desenhar esses novos protocolos, né? Então, a pandemia nos mostrou também algumas dificuldades, mas também desenvolvemos algumas saídas para isso.
Thank you very much, Dr. Favaro. There are only a few more messages. Maria do Céu de Góes, she said, excellent. I'm marveled with both of your studies. Congratulations. Luciana Capiroto said, thank you very much for your research in this area, Dr. Chammas, Dr. Alonso, especially when we think about the patients that will be beneficiaries from your work. Maria Isabel Alonso said, great, very productive, this round table. Congratulations, Dr. Alonso and Dr. Favaro, for the promising therapy. Thanks to Dr. Chammas for the talk. To Dr. Alonso, she said, your sister is proud of you.
Aí não vale.
And we have just about 4 minutes. Is there any thoughts, final thoughts, Dr. Chammas or Dr. both? 2 minutes to say your final thoughts, Dr. Alonso.
Bom, quero agradecer imensamente pela oportunidade de participar desse evento tão grandioso e peço desculpas, eu não quis arriscar ficar falando inglês porque realmente não tenho inglês bom e eu tentei apresentar a aula em inglês, mas vocês perceberam que não foi brilhante como o Dr. Roger, que fala inglês fluentemente. Mas eu tentei passar uma mensagem e foi um prazer poder estar ao lado do Dr. Roger Chammas, que tenho muita admiração. Querido. Você, Rodrigo, pela oportunidade. Foi excelente poder estar com isso. Muito obrigado.
Thank you, Dr. Alonso. It was a great presentation. Don't worry about speaking English. It was really good. Don't worry. Roger Chammas, please. Oh, sorry, Wagner, do you want to say something? Some final words? No, okay.
Eu peço desculpas que penetra, né? Eu penetro aqui na apresentação, mas eu gostaria de agradecer a oportunidade também, né, aqui representado pelo Dr. Jõao, representando o grupo. O mérito de todo esse trabalho apresentado com certeza, do Dr. Jõao, né, que conduziu, que acompanhou todos esses pacientes, de apresentar o que a ciência pode fazer, né, e parabenizar a toda a comissão organizadora desse evento e uma satisfação enorme, né, de ver o professor Roger Chammas. Tenho uma grande admiração por ele, e é uma satisfação encontrar o professor Roger e ver os trabalhos, a ciência muito bem aplicada do professor Roger, que abrilhantou mostrando as pesquisas e mostrando a aplicação, né, uma pesquisa que começa com a ciência básica e uma ciência aplicada. Isso é o mérito, né, que o professor Roger construiu ao longo de toda a sua carreira e que enche o nosso país de orgulho para mostrar que a ciência brasileira deve ser respeitada e que nós fazemos, quando se pergunta o que os pesquisadores, o que os professores, o que a universidade faz, acho que é o grande exemplo do que uma universidade pública faz no Brasil e devolver para a sociedade todo o investimento feito em nós. Obrigado pela oportunidade.
Thank you, Wagner. Professor Chammas. Your mic is off.
Agradecer a oportunidade, Rodrigo. É um enorme prazer estar dividindo a mesa redonda com o Alonso, com o Wagner. De fato, a reciprocidade existe. Eu tenho um enorme apreço pelo trabalho que eles vêm fazendo e que eu acompanho já até perdi a conta, né, Wagner?
Com certeza.
De qualquer forma, queria estender os parabéns à comissão pela visão, né? Acho que o tema que é colocado, assim, eu acho que a gente não deve colocar fronteiras entre o que é fundamental e o que é aplicado, o que é, aspas, ciência básica, o que é ciência clínica. Na realidade, existe um contínuo de aplicação, o conhecimento é o conhecimento, existe a boa pesquisa, e a boa pesquisa ela não tem outras, não deveria ter outros objetivos, né? A gente tem grupos muito comprometidos no nosso país. Queria deixar uma nota positiva, né? Acho que a gente tem uma competência instalada muito, muito boa no nosso país, a gente tem uma clínica muito boa no nosso país, o Sistema Único de Saúde pode ter problemas, mas ele funciona bem. O que eu acho que a gente ainda não tem são formuladores de política pública que nos ajudem a fazer essa transição, e eu acho que é nosso papel exigir, uma vez que a gente tem um pacto sobre o que fazermos, eu acho que a gente tem que exigir a organização de políticas de Estado que permitam fazer essas coisas fantásticas que o grupo da Unicamp está fazendo, que, guardando as devidas proporções, a gente está conseguindo fazer aqui no ICESP, juntando diferentes grupos. Em última análise, como disse algum dos ouvintes, dos participantes, a gente não está fazendo isso para a gente está fazendo isso para os nossos pacientes. Quer seja uma pesquisa fundamental, quer seja uma pesquisa clínica, de novo, não importa esse rótulo. A gente devemos nos entender como pontos de um contínuo, né? E acho que a mesa redonda tem um pouco essa função também de conscientizar as pessoas dessa nova cultura que a gente tem que abraçar. Queria agradecer de novo, Rodrigo, pela grande oportunidade de estar compartilhando com vocês esses momentos aqui. Parabéns à Unifesp pela iniciativa.
Bom, a gente está falando português, então vamos lá. Muito obrigado aos dois. Eu tenho também muita admiração pelo trabalho do professor Roger Chammas, assim, o trabalho que você vem desenvolvendo é impressionante o trabalho, e a responsabilidade que tem em estar coordenando as pautas clínicas, e CESP está no meio de tudo isso, fazendo esse excelente trabalho no meio de uma pandemia, todas as coisas que estão acontecendo. Wagner, muito bom o trabalho de vocês, parabéns, vocês estão indo para o caminho que, assim, é uma coisa que a gente precisa desenvolver, trazer isso para o público, né? A gente tem na DCG que a gente tem, tá? Então, agradeço também a todos os participantes que vieram, participaram, trouxeram perguntas e assistiram, e espero que vocês saibam que vocês são divulgadores, então tudo que vocês viram aqui vocês vão estar passando adiante, falando para as pessoas da importância da saúde pública, de tomarem cuidado, de aprenderem novas técnicas, de estarem desenvolvendo e levando as pessoas para o hospital para fazer o que deve ser feito, tá? E, claro, políticas públicas trazendo a importância de a gente ter investimento em ciência básica, ciência aplicada, seja ela qual for, porque a gente está num momento em que a gente tem que ter esse apoio não pode ser ofertado e retirado da comunidade científica. Bom, com isso eu finalizo essa sessão agradeço a todos e, então, me despeço de todos vocês. Muito obrigado.
Obrigado.
Recording stopped.
Pessoal, nós pedimos que vocês ainda continuem aí, né? Os que nos